Hello friends, today in this video I am going to discuss about the MCQs on general microbiology, and this is the last part of the series of 500 MCQs on general microbiology. So let's start the question answer session on microbiology. So the first question is, anaerobiosis can be maintained by incorporating reducing agents in media such as 1% glucose, meat particles, thioglycolate or 0.05% cysteine. Okay, and the right answer is option E. That is, all the answers are correct. So next question is, anaerobic culture media include thioglycolate medium, glucose broth, Robertson's cooked meat medium or Loeffler's medium. Right answer is option B. It is 1 and 3, that is glycolate medium and Robertson's cooked meat medium is a anaerobic culture media. So next one is, examples of transport media include Pike's medium, alkaline peptone water, Stewart's medium or Sabord's medium, and the right answer is option A. It is 1, 2 and 3. First three are the right answer about the transport medium. So these are the transport media. Okay. So move on to the next question, that is, example of enriched media include Loeffler's media, chocolate agar, Dorset egg media, McConkey's agar, and the right answer is option A. That is, first three are enriched media, that is Loeffler's media, chocolate agar and Dorset egg medium are enriched media. So TSI media is used to detect gas production, H2S production, sugar utilization, citrate utilization. Right answer is option A. First three options are correct. That is, TSI medium is used. TSI means triple sugar iron agar media. So that is used to detect the gas production, H2S production and sugar utilization. Okay. So next one is, standard method of antibiotic sensitivity by disc diffusion include Kirby-Bayer method, Luria, Delbruck method, Stokes method or slope dilution method, and the right answer is option B. It is 1 and 3. Kirby-Bayer method and Stokes method are used as an antibiotic sensitivity test. So next one is, bacterial plasmids are extra chromosomal DNA, circular double-stranded DNA, self-transferable or essential for the survival of bacteria. So the right answer is option A. These first three options are correct. So bacterial plasmids are extra chromosomal DNA, circular double-stranded DNA, self-transferable element. Okay, so move on to the next question, that is, plasmid is transmitted among bacteria by transformation, transduction, transfection or conjugation, and the right one is option C. That is 2 and 4. So bacterial plasmid transmitted by transduction and conjugation. So next one is, bacterial genes are transferred by transformation, transduction, conjugation or mutation. So right answer is option A. That is, first three options are correct. So bacterial genes are transferred by transformation, transduction, conjugation. So next one is, drug resistance in bacteria is seen with mutation, transformation, transduction, conjugation. So right one is option E. So all are correct regarding drug resistance in bacteria. So next one is, transformation requires bacteria in a state of competence. Is transfer of genetic material through the agency of bacteriophage. Is a process involves single-stranded DNA integrating into recipient DNA. Is a reversible process. So the right answer is option B. That is 1 and 3 is the correct regarding transformation. So next one is, mutation is a random event, a heritable variation, involves alteration in nucleotide sequence or a reversible process, and the right answer is option E. All the options are correct about the mutation. Okay, so next one is, true statements regarding mutation include nonsense mutation results in termination of peptide, missense mutation result in altered amino acid, suppressor mutation is reversal of a mutation, conditionally lethal mutation is always non-permissive. So the right one is option A. So first three option is the correct about the mutation. Okay, so next one is, a transduction in which a segment of donor's chromosome adjacent to the site of bacteriophage attachment is transduced is called abortive transduction, generalized transduction, chromosomal transduction, or restricted transduction. And the right answer is option D, that is only 4. So restricted transduction is the right answer. So next one is the process of transduction has been observed in gram-positive as well as gram-negative bacteria. It requires bacteria instead of competence. It involves the packaging and transfer of DNA by a bacteriophage or is sensitive to the presence of DNAs in the buffer. And the right answer is option B. It is 1 and 3. 1 and 3 is the correct answer about the transduction process. Okay, so next one is R plasmid of bacteria. R means resistance. Okay, so resistant plasmids of bacteria is similar to F factor, possess information for resistance to antibiotics, cannot be transferred to all bacterial cells, possess insertion sequence for their transfer. So the right one is the option A. So first 3 options are correct regarding R plasmid of bacteria. So next one is F plasmid in bacteria is responsible for synthesis of sex pillars, self-transfer, integration to host chromosome, transfer of drug resistance. And the right one is option A. So first 3 options are correct about the F plasmid. Okay. So next one is lysogenic phage conversion is conversion of prophage into a lytic phage, detachment of prophage from bacterial chromosome, transformation of a virulent phage to avirulent phage, a change in bacterial phenotype due to the presence of a prophage. And the right answer is option D, that is 4. So a change in bacterial phenotype due to the presence of a prophage is the right answer regarding lysogenic phage conversion. So next one is the transport media for Vibrio cholerae include Venkataraman Ramakrishnan, that is the VR medium, or bile peptone transport medium, alkaline peptone water, Carey Blair's transport medium. And the right answer is option E. So all are the transport medium used for Vibrio cholerae. So next one is special media used for the cultivation of Vibrio cholerae include alkaline bile salt agar, Monsource, gelatin, terracolate, trypticase, tellurite agar, that is the GTTA agar, or thiosulfate citrate bile sucrose agar, that is the TCBS agar, or deoxycholate citrate agar. So the right answer is option A. So first 3 option is the correct option regarding the special media used for the cultivation of Vibrio cholerae. Okay, so the next one is the test used to differentiate Vibrio cholerae biotypes include hemagglutination of RBCs, VP test, polymyxin B sensitivity test, or cholera rate reaction. Okay, so the right answer is option A. This first 3 option is the correct answer. So next one is the true statements regarding cholera treatment is patient can be treated at home. Once treatment is started, recovery is slow. Vaccine should be routinely given. Immediate treatment is oral replacement of fluid and electrolytes. And the right answer is option D. So the last option is the correct regarding the cholera treatment. That is the immediate treatment is oral replacement of fluid and electrolytes. So the next question is Campylobacter is described as gram-negative, comma-shaped bacilli, microaerophilic or strongly anaerobic organisms. And the right answer is option A. These first 3 options are correct regarding Campylobacter jejuni or Campylobacter organism. Okay, so they are gram-negative, comma-shaped bacilli and microaerophilic. So the next question is causative agent of diarrhea associated with ingestion of contaminated food or fluids include Campylobacter, bacteroids, Vibrio or Staphylococcus. And the right answer is option B, that is 1 and 3. So Campylobacter and Vibrio. So causative agent of diarrhea associated with ingestion of contaminated food include Campylobacter and Vibrio. Okay, and the last question of this series of 500 MCQs, that is the regarding infection caused by Campylobacter. So Campylobacter jejuni infection are self-limiting. Campylobacter jejuni Specimen should be incubated at 42 degrees Celsius. Special care on food and personal hygiene can prevent diarrhea due to Campylobacter, or the last option, that is infection with Campylobacter are rare in relation to Salmonella and Shigella. And the right answer is option A, that is first three option is correct about the Campylobacter. Okay, so these are the all 500 questions regarding general microbiology. Okay, thank you for watching this video.